Let me thank you all and say a very good day to you, good vector control colleagues from across Asia Pacific, and some also from as far away as Africa and elsewhere. I think there are also some people that registered from North America. Very impressive. A warm welcome to all of you. Thank you for finding time to listen in to this APMEN Custard University online course on insecticide susceptibility monitoring. This is our very first module of online training of what we intend to be an extensive series of online modules and tutorials that we will be presenting in months ahead. So this is our trial prototype pioneering effort, and we hope it's going to, we intend to expand it into something much bigger, and we will learn and improve as we go along. So just a quick note, APMEN stands for the Asia Pacific Malaria Elimination Network, and together with the Asia Pacific Leaders Malaria Alliance, APALMA, it works to support the 21 member states in Asia Pacific to reach their common goal of malaria elimination by 2030. APMEN achieves much of its work through its 3 working groups, namely the VIVAX working group, the Surveillance and Response working group, and the Vector Control working group, which is the one that has worked with Custard University to produce these courses. Custard University is one of the 3 biggest universities in Thailand, and its entomology department is widely respected throughout Asia Pacific as a center of excellence. Custard and the Department of Entomology hosted the intensive 2-week malaria vector surveillance for elimination course back in 2019, and it did such an excellent job that APMEN contracted them now to prepare an online version, of which this module is one part. The department is led by the highly respected Professor Tirapap Charionviriapap, and we have the privilege today of having Prof. Tirapap and also Dr. Jiro Nararak, who led the team putting together the video courses, to be present with us today to respond to questions that any of you may have on the tutorials that will be shown. By the way, as far as I understand from my good colleague Wulan, we have about 182 people who registered for this course. It's mind-boggling. It's wonderful to see this response. And thank you, sincere thank you to you all for your interest and for registering. We'll see how many actually participate, but at least 182 people registered. We will have 2 tutorials today. The first one will be a PowerPoint, and that's about 14 minutes long on insecticide resistance mechanisms. And the second is a video on the WHO tube test for detection of resistance. I can't remember how long that one is, but I think it's about 8 minutes, something like that. We will have a very short poll after each tutorial. So we have the tutorial and then 2-3 minutes for a little poll. And we ask each of you to please complete that poll before we move on to the next tutorial. Your answers help us to understand what did we do right, what did we do wrong, where can we improve, what do you think of the particular tutorial. It helps us to put on a better product next time around. And then at the end of both tutorials today and also tomorrow, because we'll have 2 more tutorials tomorrow, there will be a question and answer session that deals with both tutorials that you will see today, and then same tomorrow. All audience members are muted and video off, but we ask you please to exchange notes and chat with each other and put comments in the chat box. But please put your questions in the Q&A box. We are not going to go through the chat box for questions. So please put your questions in the Q&A box. And if you see someone else's question and you think it's a good one, you can actually upvote it so it goes higher up the list, and we are more likely to read out that particular question. Please note that all of this is being recorded, and we will upload it onto the OREEN platform very soon. Look on your screen and you'll see the website www.oreen.org, or the OREEN URL. I think we'll do it within about 48 hours, but it'll be up there very quick and soon. Please also note that we will be offering a certificate to each of you for attending, but you must complete a short multiple choice questionnaire at the end of the 4 tutorials, and you must score at least 70% or more to qualify for the certificate. So please, you need to concentrate and listen during the tutorials. They're not going to be very hard or difficult questions, and it's 10 questions that we will be asking you in multiple choice format to answer. But we can't issue you a certificate and you haven't proven that you've at least listened to the tutorials and you retain some information. So we ask you to do that multiple choice questionnaire, and then we'll send you a certificate of a satisfactory participation. Finally, let me also say thank you to our competent backroom team that quietly makes sure everything goes well with the course presentation. These are Dr. Tin Cho Thu, our APMEN VCWG program coordinator here in Bangkok, Dr. Ponsi Hein, who's the APMEN program coordinator in Singapore, and then Mrs. Shobicha Wulandari, Wulan, who is technical officer for Malaria Consortium in Bangkok. I also need to say a big thank you to the Malaria Elimination Initiative of the University of California, San Francisco, who provided the funds to enable the course to be produced, and also the United States Centers for Disease Control and Prevention for funding the presentation of this course. So I think enough talking now. Let's go on to tutorial 1. Wulan, would you mind starting tutorial 1 for us? Remember, this is going to be about 14 minutes, and then the short poll after that. Let me just find the laser pointer here. So, hello everyone, welcome to our tutorial on insecticide resistance modes of action. This is going to be a quick introduction to some of the different kinds of insecticide resistance, how some of them work, and what you should be doing about it. So for more than a century, vector control has been the most important strategy in the fight against malaria, and almost as long for various arboviruses. There are different methods of vector control, some mechanical or physical, such as emptying water containers against Aedes larvae, biological methods such as fish that feed on mosquito larvae, and then chemical insecticides. The most widely used method has been chemical insecticides for many decades now, especially in insecticide-treated bed nets, and also residual spraying. However, long-term and frequent use of the same insecticide can result in the adaptive selection for resistance. And this slide shows how initially an insecticide targets the susceptible proportion of mosquito populations, which make up the majority. These blue mosquitoes here are the susceptible ones. But usually there are a few individuals that are genetically resistant, or they will emerge over time as you keep using this insecticide. A mutation will result at some stage or other in individuals that are increasingly showing resistance, and they will survive. And these few individuals will produce offspring that are also resistant to the insecticides. And so over several generations, more and more of the population becomes resistant, and eventually only the resistant strain survives to reproduce. There are 4 main categories of insecticide resistance. The first one is metabolic resistance. There's also target site resistance, cuticular thickening or reduced penetration, and behavioral avoidance. This slide shows the main classes of insecticide we mostly use in mosquito control. And these are the carbamates, the organophosphates, the pyrethroids, and the organochlorines. So these insecticides kill mosquitoes by interfering in the transmission of nerve impulses. When stimulated, here's the surface of the body, for example, either by tasting something, seeing, smelling, feeling, or whatever, that nerve cell sends an impulse to other neurons, to their equivalent of a brain or an effector organ, such as a muscle, along a chain of other nerve cells. In each nerve cell, the impulse has to travel along these cord-like extensions called axons. They're all part of the same nerve cell, a single neuron. And then the impulse goes along the axon and it reaches a synapse, a junction, a bridge that links it with the next neuron. Don't worry about these names, just try to get the general idea. So the nerve impulse has to travel along a chain of other neurons to reach the brain or the effector organ, and they do so by traveling along these axons and the synapses. Some insecticides interfere with nerve impulses processes along the axon, this region over here, and some interfere with the process at the crossing of the bridge, the synapse, in this region over here. The pyrethroids and the organochlorine insecticides interfere with little sodium channels that open or close. This is the outside, this is the neuron over here. The outside, just outside, this is the cell membrane, and then the inside of the nerve cell. And you get these little bridges, protein bridges. So some pyrethroids and organochlorine insecticides interfere with little sodium channels that open or close on the axon membrane. These sodium channels regulate the passage of electrically charged ions across the cell membrane, and that controls the signal being passed on. There's an electrical difference between the outside and the inside of the neuron, negative and positive, and these channels open to allow ions to flow. It changes the electrostatic difference, and that triggers the passage of a nerve impulse. Some pyrethroids or organochlorines either prolong the opening of a channel, which results in excessive firing of the stimulus and hyperexcitation, and some others block the channel, and this leads to paralysis. Carbamates and organophosphates impact at the nerve synapse, this bridge between two different neurons. When an impulse reaches the nerve end, it releases acetylcholine neurotransmitter into the gap. The acetylcholine is in these little vesicles. It receives a stimulus. The neurotransmitting acetylcholine gets released into the gap. It triggers an impulse in the neighboring neuron. But then once this acetylcholine has been released and it's triggered the next one, then you need an enzyme, acetylcholine esterase, to degrade the acetylcholine in the gap. Otherwise, it just keeps on triggering the adjoining nerve cell. So carbamates and organochlorines bind with choline esterase, so the acetylcholine stays present and it keeps triggering the nerve impulses, and that leads to paralysis and death. So here are the major mechanisms of insecticide resistance. In the case of metabolic resistance, the top part of this slide over here, resistant insects detoxify or destroy the toxin faster than susceptible insects. or quickly rid their bodies of the toxic chemicals, toxic molecules. Here you can see how the detoxifying enzymes have either modified, altered the structure of the insecticide molecule or broken it, so it is no longer able to bind with the target proteins. Metabolic resistance is the most common mechanism of insecticide resistance. Insects use their internal enzyme systems to break down insecticides, and resistant strains may possess higher levels or more efficient forms of these enzymes. And in addition to being more efficient, these enzyme systems may also have a broad spectrum of activity. In other words, they can degrade many different kinds of insecticides. Metabolic resistance mechanisms have been identified in mosquitoes against all major classes of insecticides currently used for vector control. So against organochlorines, organophosphates, carbamates, and pyrethroids. In the case of target site resistance, here at the bottom, over here, the particular site within the insect body where the insecticide acts is modified to prevent the insecticide from binding or interacting, thereby reducing or eliminating the toxic effect of the insecticide. So the insecticide is still exactly the same, hasn't been denatured, degraded, broken down or anything, but the target site has changed, so it doesn't work anymore. But remember, we mentioned there are also other forms of resistance. Penetration resistance over here is when such resistant insects absorb the toxin more slowly than susceptible insects. It occurs when the insect's outer cuticle develops barriers which can slow absorption of the chemicals into their bodies. Cuticular thickening is also a method, so that the insecticide has to travel further, and it gives more time for enzymes to detoxify, etc. Behavioral avoidance or resistance happens when insects change their behavior to avoid the toxin. For example, Anopheles populations that change from being mainly indoor feeding to become mainly outdoor feeding, genetic selection, and bite early evening instead of late at night, which means that they are less likely to come into contact with insecticide-treated bed nets. Cross-resistance occurs when a resistance mechanism, which allows insects to resist one insecticide, also confers resistance to another insecticide. Cross-resistance can occur between insecticides from different chemical classes. Multiple resistance occurs when insects develop resistance to several compounds by expressing multiple resistance mechanisms. The different resistance mechanisms can combine to provide resistance to multiple classes of products. So here's the most important point of this presentation. Insecticide resistance management is an extremely important task of any national malaria control program or vector-borne disease control agency. It can mean success or failure of your disease control program. Insecticide resistance management is usually achieved by one or more of the following approaches. Firstly, rotations. Rotational strategies are based on the rotation over time of two or preferably more insecticide classes with different modes of action. And so you will use a pyrethroid this year, but next year you use a carbamate, etc. For mosaics, here you can use two different kinds of insecticides at the same time, but in different places. So for example, you can spray one part of a village with one type of insecticide and the other part of the village with another kind of insecticide. This helps to reduce the chance of resistance developing, or at least slows it down. Mixtures A mixture is defined as the simultaneous use of two or more insecticides of unrelated mode of action. And then finally, combinations. Here you use different kinds of vector tools against the same mosquito population at the same time. The mosquitoes may escape contact with the one type of a vector control tool, but then they get hit with the other kind. For example, using long-lasting insecticidal nets and also indoor residual spraying. But they must not contain the same insecticide. If you use the same insecticide in the nets and IRS at the same time, you simply increase the selection pressure for the mosquitoes to develop resistance. So you have to use a combination of different insecticides. But of course, it's going to cost a lot more. So there's that disadvantage. In the remaining 3 tutorials, we are going to be looking at 2 ways to monitor insects' susceptibility to insecticides or development of resistance. These are the WHO filter paper tube test and the CDC bottle bioassay. In the final tutorial, we will discuss the cone bioassay, which checks on the residual efficacy of insecticide present on treated surfaces. And then you can read more about insecticide resistance at these websites shown below to learn more about what we discussed in this particular PowerPoint. We're going to refer you to other websites as well in the video presentations. So thank you so much. Hope you learned something. Hope you enjoyed it. Okay, that's the end of our very first tutorial. I hope you didn't find it too slow or too long. Let us know in the chat box. Tell us what you think. But may I ask, Wulan, can you make the poll? Can you make the poll available now? And I'm going to ask each of you in the audience, please, will you just fill in the poll for us? It'll take you 2-3 minutes. It's not very long, 5 or 6 questions. And then we'll move on to tutorial number 2. So I'm going to keep quiet so that I don't distract you. And if you can just fill in the poll, please. We're approaching about 45% of people have completed the poll. Please, as many of you, please complete the poll as you can. Thank you. Maybe on 60% of you now. %. I'll give it another minute or so. %. Let's see if it sticks. 76%. %. 80%. All right, if you're still busy, please continue. But I'm going to start talking now again, and very soon we're going to move to tutorial number 2. So tutorial 1 is the only PowerPoint presentation we have. The next 3 tutorials, the remaining 1 today and tomorrow, same time, there will be 2 additional tutorials, tutorial 3 and 4. Those are all video based. As mentioned, we're going to move on to tutorial number 2 now, and then we'll have another short poll. And then after that, we'll have question and answer session. Please don't put your questions in the chat box. Please put them in the Q&A box if you don't mind. Thank you so much. Thank you, everyone. We're going to move to tutorial number 2 now, please. I see Dr. Pradeep, and I think I also saw Dr. Rachadawan. Wonderful to have you present. Welcome, Dr. Pradeep. Welcome, Dr. Rachadawan. Hello everyone. In this video tutorial, we are going to talk about the World Health Organization Insecticide Susceptibility Test, also known as the WHO Tube bioassay. But before we do that, let me mention as background that the near ubiquitous and long-term use of insecticides for mosquito control have contributed to the development of resistance within target mosquito populations. In May 2012, WHO launched the Global Plan for Insecticide Resistance Management in Malaria Vectors, also known as GPIRM, which sets out a comprehensive framework for action in 5 key strategic pillars. One of these key pillars is entomological and susceptibility monitoring. The WHO susceptibility test for adult mosquitoes is a widely used and internationally accepted tool to evaluate the insecticide susceptibility status of a mosquito. This bioassay is a direct response to exposure test. In other words, what is the survival response of the mosquito after being put in contact with an insecticide? And it measures mosquito mortality to a known standard concentration of a given insecticide. It can be done either as a discriminating concentration to see if there are signs of insecticide resistance, or as intensity concentrations to see how strong the level of insecticide resistance is, or using a synergist insecticide to determine the underlying cause of insecticide resistance. So you can perform assays for insecticide susceptibility in 3 ways. Susceptibility tests for determining phenotypic resistance. In other words, is there any visible physical evidence of resistance? Secondly, susceptibility tests for determining intensity of resistance. In other words, how strong is this resistance? And then thirdly, susceptibility tests for determining resistance mechanisms. So let us explain a bit more. Firstly, again, about susceptibility tests for determining phenotypic resistance. The concept of discriminating or diagnostic concentration to see if there is any evidence of resistance is done by using dosages twice the concentration which normally results in 99.9% mortality within 60 minutes in susceptible strains of the mosquito species being tested. So you expose your wild strain of test mosquitoes for 60 minutes to twice the dose of insecticide that would normally kill 99.9% of a susceptible strain of mosquitoes. And the purpose of this method is to detect the possible presence of insecticide resistance in mosquitoes or other disease vectors. Okay. Then we look at susceptibility tests for determining intensity of resistance. These tests are designed to use selected insecticides at 5 to 10 times the discriminating concentrations used to detect insecticide susceptibility in adult mosquitoes. This is a stepwise test that provides information on the intensity or the strength of a resistance, if any, and it helps us to understand the operational value of the insecticide in the field. If there is only low intensity of resistance, maybe it is still okay to use the insecticide. But if the resistance is strong, very intense, then the insecticide has no more value for use. Susceptibility tests for determining resistance mechanisms. This concept is to determine the underlying mechanism resulting in insecticide resistance. Before moving to the actual assay, let's have a look at what equipment you will need to conduct these assays. Holding tubes and exposure tubes. Mosquitoes, 70% ethanol, cotton, sheets of clean white paper, 12 centimeters by 15 centimeters, steel spring wire clips, aspirator, slide, forceps, slide unit, screw cap. To make sure that the equipment is clean, we wipe the cylindrical tubes with cotton wool soaked with 70% ethanol. Note that all these materials can be bought from World Health Organization, WHO. Also note that the tubes are separated by color. Green dotted are the holding tubes, yellow dotted are the control tubes, and red dotted are the treatment tubes. When all materials are ready, we can proceed with the assay. First of all, roll 6 sheets of clean white paper, 12 centimeters by 15 centimeters, into a tube and insert each into its own green dot holding tube. So you have 6 tubes, each with a rolled up sheet of paper. Next, secure the paper into position against the wall of the tube using a steel spring wire clip. The slide unit and screw the cap to the tubes. So you end up with 6 tubes. The first 2 tubes are assigned as control tubes, and the remaining 4 tubes are assigned as treatment or exposure tubes. Now gently blow 20 to 25 active female mosquitoes into the green dotted holding tubes through the filling hole in the slide unit. Once the mosquitoes have been transferred, the slide unit is closed and secured with a piece of cotton wool, and the holding tubes are set in an upright position for 1 hour. After 1 hour, any moribund or dead mosquitoes are removed and replaced. Now we prepare the treatment and control tubes, the actual exposure tubes. Firstly, insert 1 oil-treated sheet of control paper into each of the 2 yellow dotted tubes, ensuring that the label of the paper is visible on the outside of the tube. Then secure the paper with a copper clip and close the tube with a screw cap. The red dotted treatment tubes, the exposure tubes, are then prepared in exactly the same way, using the insecticide treated paper. Finally, the empty exposure tubes are attached to the vacant position on each of the 6 slides. When the 1 hour waiting period for the holding tubes has expired, now you can proceed with the following steps. Attach the exposure tubes to the free end of the sliding unit. Open the slide unit, then gently blow the mosquitoes from the holding tube into the exposure tube. Once all the mosquitoes are in the exposure tube, Immediately close the holding tube and carefully set them aside in a protected position away from wind or excessive light or disturbance. To prevent the slide from accidentally moving, we usually insert a cotton wool plug into the hole to lock the slide. The mosquitoes are kept in the exposure tubes, which are set in a vertical position for a period of exactly 1 hour. These make sure the tubes are placed in reduced lighting or covered with cardboard discs to reduce light intensity and to discourage test mosquitoes from resting on the mesh screen lid. When the 1 hour exposure period is completed, firstly, count the number of mosquitoes knocked down, in other words, those that are moribund or dead, and record that number in the results form. Then, transfer the mosquitoes out of the exposure tube back to the holding tubes. Detach the exposure tubes from the slide units. And then place a wad of cotton wool soaked in 10% sugar water on the mesh screen end of the holding tubes so the mosquitoes can feed. The cotton wool must not be too soaked. The sugar solution must not drip and drown the mosquitoes, only well moistened. The mosquitoes are kept in the holding tubes for 24 hours. During this time, it is important to keep the holding tubes in a shady, sheltered place in the laboratory or insectary. Temperature and humidity should be recorded during the recovery period. And then at the end of 24 hours, the number of dead mosquitoes is counted and recorded. Just so you are aware, a mosquito is classified as dead or knocked down if it is immobile or unable to stand or fly off. So here is some guidance on interpreting the results of the WHO susceptibility test as provided by WHO in 2013. If 98 to 100% of your exposed mosquitoes are dead or knocked down, it means the population of mosquitoes is still susceptible to the particular insecticides you used to test them against. A mortality between 90% and 97% indicates the existence of resistance, but further investigation is needed to confirm it. If mortality is less than 90%, this is confirmation of the existence of resistant genes in the test population. It is then necessary to test additional samples at the 5 times and 10 times concentrations to determine the intensity of resistance. Further investigation of the mechanisms and distribution of resistance should also be undertaken. If mortality in the control bottle is greater than 20% at the end of the bioassay, the results of this particular run should be discarded and the WHO susceptibility bioassay should be repeated. Abbott's formula can be considered when control mortality is less than 20%. Please read up about Abbott's formula if you have control mortality approaching 20%. Our final guidelines for this WHO insecticide susceptibility bioassay are about washing materials. Firstly, wash each tube with soapy water and rinse thoroughly with water at least 3 times. Tap water can be used for this step. Leave the tube to dry completely at room temperature or in the sun. To assure that the cleaning procedure is adequate, introduce some susceptible mosquitoes into a sample of recently washed and dried tubes. Mosquitoes should not die at any greater rate than a sample of mosquitoes in a known clean tube. If they do, repeat the washing and drying procedure. You can read up more on this subject by accessing the URLs shown on the screen. Well, there we are. Thank you so much to our team from Kasetsart for the production of those videos. Thank you to our audience for your patience. I've already picked up one error in that video, and I'll, as I said, this is a prototype trial production, and we need to refine the materials. But I also want to make the comment, I don't want you to get the impression that you can just look at the video and you'll know all there is to know about this particular WHO tube by Assay method. It's best to have the manual, the written manual, available for reference, and then to read the WHO guidelines on how to do the test and use the video to help you interpret what is being said. So you get a written version and you get a visual version that helps you to understand how to conduct this test. I think that's the best way to go about it. Very often you read a set of instructions, but it helps to visually see how somebody else is doing it. And I think this is probably the best way that these videos can help you, is as a supplement, not as a replacement, but that the two should be used together. So I'm going to ask Wulan if you wouldn't mind just putting up the poll again so our participants can have a look, can give us a comment on the video tutorial. I'll give you a few more minutes to do that, and then we go to questions and answers. Thank you. Okay, we're on 80% and we seem to be stuck on 80%. So that's probably as good as we're going to get, 82%. All right. So if you are busy finalizing your poll, please continue. But I think let's move on to the questions and answers. So some of you did post some questions in the chat box, and it complicates it a little bit. We're going to use primarily the Q&A. So Wulan, I'm not sure if I am, but I get the impression that, the questions from tutorial 1, we may have lost by moving to tutorial 2, and that's a bit of a problem. So, sorry, I wasn't aware of this, but those of you that had questions from tutorial 1, and they may not show up here in the Q&A over here, may I ask, and I apologize for that, if you wouldn't mind just writing down your question again in this Q&A box, because I don't know how to go back to the Q&A of the previous session. So let's just see here. So when a population is resistant, here's a question from, the name seems to be cut off, Intan Ishak. When a population is resistant to insecticides of different modes of action, say for example, pyrethroids and organophosphates, is it considered as cross-resistance or multiple resistance? Prof. Tirapap, do you want to try that, responding to that question? I think you may be on mute, Prof. Tirapap.
Can you see me, Professor Leo?
I can't see you, but I can hear you.
Okay,
So
I don't know what happened now, but the questions, can you repeat the questions?
Okay, just check if you click on your Q&A box at the bottom, right at the bottom of your screen, there should be a Q&A icon there. Just click on that, and the very top question from Intan Ishak, right at the top of the list, says, when a population is resistant to insecticides of different modes of action, for example, pyrethroids and organophosphates, is it considered as cross-resistance or multiple resistance?
Okay, if insecticides share the target site or the pathway, this we can consider this is the cross-resistant. For example, DDT and pyrethroids. This is for example. But if they did not share any mode of action, the same mode of action, this we consider this is multiple resistant. Is that clear?
Yes, no, I think you explained that very well. Thank you, Professor Tirapap. That's well explained. And I think, Agye Kumi, your question is basically exactly the same. So I think Professor Tirapap has answered your question at the same time. Jaipal Singh, ideally, when will one conduct an insecticide resistance management study? So, you know, I think I'm going to jump in once in a while as well, just to relieve the pressure on Prof. Tirapap and Jirod. But if I'm wrong, or if you feel you have a better answer, Jirod, Prof. Tirapap, or Dr. Rachidawan, or Prof. Pradeep, please feel free to jump in and add on. But if you are basing a vector control program that is partly or wholly based on insecticides, you should, as early as possible, already start convening a little working group to investigate and assess the options based on literature studies, based on consultations and interactions with entomologists, National Malaria Control Program staff in other countries that have similar vector control programs as you. Because sure enough, if you are using an insecticide for vector control, sooner or later you will face that challenge of having insecticide resistance. So don't wait until your problem is present. Start planning what you're going to do beforehand. So that would be my response. Prof. Tirapap, any comment there?
No, I agree with you. I think it's good when you start using the insecticide target the population. I think soon after, I think you should think about how to do the insecticide resistance study, because the cycle of mosquito it's very short and can be the offspring can carry on the gene from one generation to another generation. The quicker, the better.
Thank you. A question from Ezekiel Wahe. How is mosquito resistance distributed across the African continent? What type of resistance is common in Africa? So as a general statement, insecticide resistance is becoming more and more widely distributed across the world, not just, but particularly in Africa. And this is a major problem and a large contributing factor why, there was this strong decline in malaria case numbers between the year 2000, and then from about 2015, the numbers started reaching a plateau and declining. And insecticide-treated bed nets is our #1 vector control intervention. In fact, it's our #1 method for combating malaria, insecticide-treated bed nets. And it's the increasing resistance to these insecticide-treated bed nets that's contributed in significant or major measure to the decline in the effectiveness of our malaria control programs. The bottom line is, for indoor residual spraying, for bed nets, because humans live inside those dwellings and they're exposed to those insecticides, mostly up to now, it's been pyrethroids and in some cases DDT. Organophosphates, carbamates, and so on, a little bit more dangerous and toxic. You don't want to expose humans too much to it. So it's been mainly pyrethroids based. That's been the main insecticide being used, and that's where insecticide resistance builds up pretty quickly in pyrethroids. Prof. Tirapap, comment from you maybe?
No, I think you are good now. So I think enough information that you have discussed.
Okay. Here's a good question from O. Singh. Gerard, I'm going to pass it to you. How many times can one use the same insecticidal paper for reuse in the WHO bioassay? Do you think you can respond to that one, Gerard? If you open the sealed package and you start using it, how many times can you reuse that piece of paper?
So it's for the implanted paper used at the WHO susceptibility test, which it depends on the chemical that we use, but mostly that we treat on the insecticide, the synthetic insecticide. So we recommend that we use the same experiment that we tested. Like we go to the field and that we use for 4 replication or 5 replication, and we keep, and also it depends that where we keep the implanted insecticide. Like if we keep in the good condition, like in the freeze or in the 4 degree, it says that they can maintain the insecticide for 1 or 2 months. But if the insecticide that's keep outside that with warm temperature, so we recommend that this only once a time.
May I add up the answer, Professor Leo? Actually, there is an expire date also, so maybe they have to check to be sure that the expire date is still okay.
Excellent. There's a full response. Good guidance there. So thank you for that. Here's a name, I'm not sure I'm going to get it right, from Viran Jan Yulu Chenuru. After 60 minutes and 24 hours recording of data, how to calculate knockdown and mortality? Any formula there? So that's pretty straightforward, and it's in your, that WHO manual. It's a very straightforward formula. So if you have 100 mosquitoes and 2 out of the 100 are dead, that's still above 98% susceptibility. So your population is still susceptible. So it's very simple. And I think the cutoff points were another 10% above 90%. etc. So have a look at your WHO manual, and the URL was shown at the end of the video there, or you go to Google and you type in WHO tube test, WHO insecticide susceptibility test, some combination like that. It will come up with that manual, print it, have a read through that. Don't rely only on this video. You try and do both together, and all of the guidelines will be there. Do you want to comment on that, Prof. Therapup?
No, I think this is very straightforward one that if you know the number of knockdown, number of mortality, and then you can calculate back to the total mosquito use. I think this should be not very difficult for everyone, I think.
Okay. No, I agree. Thank you. Mohammed Nazrul Islam, mortality 90 to 97% indicate existence of resistance, and less than 90% indicate full resistance in the exposed population. I'm not sure I understand that question. It gets a bit complicated for me. I wonder, Dr. Pradeep or Jirod, would you mind reading that question, see if you can understand it, and maybe respond in a minute or two? I'm going to move on.
Which one?
It's the one from Mohammed Nazrul Islam. It's about the number 4 or 5 question from the top. Mortality 90 to 97% indicate existence of a resistance. Has somebody got an answer to that, or maybe think it through and then we can come back to it? How many times same insecticide paper can be reused for WHO? I think we've responded to that one already. This Q&A list jumps up and down, so I lose my place. For a malaria eliminated country, how important is it to test for all classes of insecticide? Prof. Therapup, for Sri Lanka, where elimination has occurred already, should they test their, presumably it's good practice once a year maybe to conduct, as part of your routine entomological surveillance, once a year conduct insecticide resistance status. How would you approach that issue in a country where elimination has already been reached?
It's a very good question. I think I agree with you. If they can do at least once a year, so by this way they can monitor what happening in the field, just in case if you have enough mosquito to test. I think we recommend that to do it.
Yeah. Yes. No, I agree. Dr. Pradeep, I'm going to ask as a National Malaria Control Program manager for you to respond as well. But it just makes sense, if you've eliminated malaria, China, Sri Lanka, several other countries, it doesn't mean to say you've got rid of the Anopheles mosquitoes. It just means that you've got rid of parasites entering and establishing themselves, and you're preventing local transmission of the parasites. But the vectors are still there, and you will forever remain susceptible, vulnerable, receptive to reinfestation as long as there's malaria still somewhere in the world, because those parasites, somebody with malaria can fly into your country and reinfect the mosquitoes that you still have. So, and therefore it's good practice. You can't, once you've achieved malaria elimination status, you can't disband your National Malaria Control Program. Those activities have to continue maybe at a scaled down pace, but you have to retain those staff. You have to retain the ability, the capacity to respond if all of a sudden you have reinvasion of malaria into your country. And as part of that process, you need to monitor your vector populations, and part of that surveillance involves checking once in a while what is the resistance status of these mosquitoes in my country, so that if suddenly we start getting malaria cases popping up caused by somebody that's flown in from India into Sri Lanka, and they had malaria asymptomatic and they infected local mosquitoes, and now I have to respond and go and implement urgent remedial action in the outbreak area. What am I going to spray? You know, I need to understand how susceptible is the population. They're still susceptible to the insecticide that I want to use. So it's good practice to continue with surveillance. Dr. Pradeep, do you want to venture an answer maybe? I'm not sure if you're there, Dr. Pradeep.
Totally, I think we lost Dr. Pradeep.
You've lost him. Okay. Okay. Thank you, Dr. Pawan. Here's a question from Nirmal Subasinghe. What are the most suitable species to do susceptibility test? Most susceptible species to do susceptibility test is your main vector where you are, where your malaria transmission is taking place in your country, the main vectors. You need to do susceptibility assays on those species. Dr. Tirapap, comment? So the question was, what are the most suitable species to do susceptibility test?
I think it depends on the countries. If you have minimus, not minimus in the country like Cambodia, Thailand, so this is the one that you have to do on. I don't know if I answered the question. What suitable species? It depends on the availability of mosquito species, and we have to think about the main vector, the one that's the very important vector.
Absolutely. So, you know, if you have one species that's very abundant, but it doesn't really bite at humans, and you have another species that's not quite as abundant, but it's very effective and very capable of hosting and transmitting the Plasmodium parasites, it doesn't help for you to do the susceptibility test on the species that is common, but it doesn't bite humans. You need to do the tests on the species that are transmitting the malaria parasites. So you need to understand which are the vectors responsible for transmitting malaria in your country and in that particular area, and then you need to do susceptibility tests on those species. Sorry, moving on. DDT is being used in IRS for many years, so we can expect that resistant populations are already present. Are any insecticides being used? Any insecticides are being used currently in IRS? Why are we still using DDT for so long? DDT is a very effective and long-lasting and cheap insecticide. It's inexpensive. It lasts for multiple months. You want to use an insecticide that is good at killing the mosquitoes. You want to use an insecticide that is relatively inexpensive, and because national malaria control programs never have enough money, so you have to make your dollars stretch as far as possible. Thirdly, it doesn't help to have an insecticide. Some insecticides are very good, but they're only effective for about a month or 2 months. So then it means the wet season lasts for 6 months, but this insecticide is only effective for about 2 months. It means, you know, every 2 months you need to go and respray. It becomes very expensive. So ideally you want an insecticide that lasts as a residual life for a long time, that will help give control throughout the wet season. So that is why DDT is very popular in some African countries. It has a bad reputation. It's actually banned. It's got Stockholm Convention, but it has approval for malaria control purposes because there's no suitable alternative, or you know, it's just difficult to replace DDT easily at this stage. So that's why DDT is still in use. Giro, do you want to say anything about DDT maybe?
So DDT is have been banned mostly in Thailand already, and yeah, because like they widely used it before, so they have like, and also they have like some the same of like mechanism resistant with the other chemical. So that's why we have found some resistant with the other chemical that the same with mode application with DDT.
Thank you, Jiro. Here's a question from Srisant. Should we maintain conditions of less than 30 degrees centigrade during the test and recovery period? Yes. You need to, I forget, but it's, you know, ideally 25 to 7 degrees centigrade. What you're trying to do is you're trying to allow conditions that will optimize survival of the mosquitoes. So that if there's any mortality, any death, it's likely to be due to the insecticide, not because the insect became overheated, dehydrated, because of the external environmental conditions. You want to make sure that the mosquito is comfortable, it's got sugar water to drink, it's not cold, it's not hot, it's not exposed to too much direct light, so that if it dies, it's likely to be the insecticide. And that is why you try and maintain, and also the humidity. It's important to keep relatively high humidity as well, preferably above 70% relative humidity. Lao Ming, what is a good method on how to carry out sampling mosquitoes for test and can declare that the locality is confirmed with insecticide resistance? So, Prof. Thierepap, so explain to us a little bit about those mosquitoes that you want to use for your insecticide resistant status to check on whether they are resistant. Obviously, you're not going to be using mosquitoes that have been maintained in the laboratory insecticide for 30, 40 generations. You want to check if the mosquitoes out there in the wild where malaria transmission is taking place, you want to check the susceptibility status of those mosquitoes. So how do you go about collecting a sample of mosquitoes for testing? Do you catch wild females and then you get them to lay eggs, or can you collect larvae and rear them to adults and get an F1 generation? How do you do that, please?
It's a very good question. I think, Giro, you can answer this question. I think you...
Okay, thanks, Giro.
Yes. Yeah, so yeah, it's really important to monitoring the mosquito resistance, especially in the field, because we like to know that area they have the mosquito resistant to the chemical or not when we use the insecticide in the area. But the anopheles, so they have like many situations because like anopheles, sometimes like if it depends on the situation, if we can catch the mosquito by like human landing or carbamate, and if they have enough, so then we can use the mosquito at the F, we call like F0 to be tested. But in that step, we have to careful like because sometimes we got like mosquito is like fed already or unfed. So we have to like separate the status of mosquito, because like we would be like export in different result between fed and unfed mosquito. And probably that we can estimate the age of mosquito in that population by do like pallas, non-pallas, that we can know that which age of mosquito, because also the age of mosquito is also have the impact of the result also. But sometimes if we would have like not enough number to test in the field, so that we can bring the wild caught into the lab, and then we link them, and then let them go to the outspring for the F1s to make it to increase more population. But like in our lab or in the publication that they are allowed to use the outspring is not more than F3, because like it's like after that they are become like laboratory stain, or is not like related to the field behavior of field population.
Thank you, Gerard. Wulan, I see you wanted to respond to that question as well. Wulan, do you want to say something on that?
No, sorry, just I'm going to, tick done. Just, yeah, that's all. No, I didn't want to answer the question. I just want to tick down so it can go to the answer.
Okay, I
box.
see. Yeah.
Yeah.
All right. So now I've lost my place here. Okay, here's another question. According to the WHO guideline, we should use F1 generation only. Why? So I think Gerard has already partly answered that question, but it's basically, you want to standardize on the age of, the response of to the insecticidal strips differs across the age of young mosquitoes respond differently to old mosquitoes. So you want to standardize on a particular age group, and usually that's 3 to 5 day old females that have not had a blood meal before the test. It was another question that I saw in the first tutorial, is that you interfere with you influence the result of the insecticide test if you use females that are blood fed, as they will respond differently to females that have not blood fed. It's always good to standardize on females that are F1 generation. And as Gerard pointed out, if you start going to F3 or F4 or F5, then you start talking about this being interbreeding, and it's no longer a natural wild population. Now you start having a laboratory adapted population of mosquitoes. So F1 is normally the standard population. You know how old these mosquitoes are, and you would choose F1, the first generation of offspring from wild mosquitoes. F1 means the first generation of offspring from wild caught, or whatever source of mosquitoes you are using, but it's the first generation of offspring. And you use females that are 3 to 5 days old. So it's a known population. So is it, so here's Kapi Raj, is it same procedure if all types of chemicals in the bioassay test, and what type of collection is good for tube bioassay test? I'm not sure I understand that question. Gerard or Prof. Tirapap, do you understand the question? I'm not sure I understand it.
What type of correction? I don't know. I think maybe maybe he asked about I don't know if the condition of mosquito or not. For example, they collect mosquito from the cow or human landing collection. So what type collection? I'm not quite sure.
Yeah. So I think maybe this question partly answers or addresses the same question from Rinzin Namgay. Is it really necessary to find abdominal status or physiological status of samples before the test? In other words, is the female blood-fed or not? Does it really matter, Gerard?
Sorry?
Okay, it's the question by Rinzin Namgay, and the question is it really necessary to know the abdominal status or the physiological status of the mosquitoes before doing the test? Why is it important that we don't use, why do we insist, no, the females must not have had a blood meal?
Let me answer this question. May
Okay.
I answer these questions?
Sure.
Actually, in the field, it's quite difficult to find out the physiological status of mosquito, because we collect mosquito that it may be non-parasitic. If you want to do this, we have to set up a small lab and do dissection. But actually, when we doing the insecticide resistant in the field to collect mosquito, and you have to record them with the blood fed or non-blood fed and do the test. This is the very straightforward, except if you want to know the age. So you have to do more on dissection.
Yeah, so I mean, it comes down to what I said earlier, is that you want to, if you want to compare susceptibility of one population to another population or within the same population over time, you need to be able to compare apples with apples. And therefore, you want to use standardized samples of mosquitoes. So they must be known age. They must be preferably all females, all between 3 and 5 days old, and none of them must have been blood fed before you do the test. So you standardize on the physiological status, the age and nutrient status and everything of the females that you will expose to the test. So that is why, but Prophtheria pup, I think, is going to the parent generation when you collect the mosquitoes. But anyway, hopefully somewhere between that, you will have got to the answer that you were looking for. So here's a question from Shweta. How can a blood meal affect insecticide resistance? Somebody willing to try and answer that one?
How can blood meal affect to insecticide resistant?
Yeah. The insecticide resistance test. We say we prefer that the females we test must be 3 to 5 days old, and they must not have had a blood meal before testing. Why do we say that, Girod? Why do we say we don't want blood-fed females to be part of the test?
Like if mosquito is already fed, so we have when we compare with the control, if in the condition that is based my experience that in the control group with the fed mosquito, is like they mostly easily die in the tube more than the unfed one. When we use the fed mosquito fed to be used in the susceptibility test. Did you get my?
Yes, no, thank you. Sorry, I'm reading another question here. Thank you, Gerard. We can't go, okay, I think we're kind of getting towards the end. Here's a question from Stephen Mwambo. I'm not sure I understand the question. See if you understand it. Can the full susceptibility different mosquito strains demonstrate variation, knockdown, or mortality, given other factors are constant? For example, same samples, bioassay procedures, testing conditions. So, if I understand correctly, Prof. Jirod, if you have an F1 generation of the same species, and they're all females, they're all 3 to 5 days old, none of them have had a blood meal, they've all been at 75% relative humidity at 26 degrees centigrade, and you expose them to insecticidal paper, will there be variation in knockdown, mortality, or response within the different individuals in the sample? Will they react differently to the insecticide they are being tested against? And I think that's the nature of biological variation, that within any population, even offspring from the exact same parents, there will always be a difference between individuals, genetic differences. Unless you are identical twins, no two offspring are exactly the same. And for that reason, you will get different responses within limits. You'll get different responses within individuals to the same set of challenges. So my response would be, yes, you might get difference in response to the same test, but it's not going to be a 10% difference in mortality. It's going to be between 0 and 2% difference. So, you know, I think any variation will be small enough that it doesn't influence your overall interpretation about the insecticide susceptibility of the population. Remember, you're not basing your test on an individual. You're basing your interpretation of the results on a sample of multiple individuals. Prof. Tirapap, any comments?
No, I don't have no comment, but I agree with you. I think this is in the biological concept. I think it should actually the variation between individuals. We should have something that happen when we do the test.
Payush Singh, how can we assess the resistance in wild mosquito directly from the field area? Is it suggested? I think we spoke about this. You know, if we catch a wild mosquito, we don't know how old it is. So, you know, we can't standardize. We catch 100 females in the wild. You don't know unless you go and dissect it. But if you've dissected it to establish how old it is, it's dead. So you can't test it for insect. So that is why we collect wild females, we get them to lay eggs, and we test the F1 generation. It's 1 generation difference. There's no significant difference there's no significant difference. There may be some difference. Remember, we said there's no two individuals, unless they're identical twins, are exactly the same. But the difference is minimal. But it's always better to try and achieve that standardized sample of individuals. So the closest you can get to your wild population is the F1 generation, for the reason that you don't know the age or other physiological status of the wild mosquitoes. Prof. Tirapap, Jirod, any comments on that?
Yeah, I agree with you. Using the wild population, you cannot know the age of mosquito, but if you use the F1, F2, you can manage the standardized system and you can have the similar individual for the taste.
Yes, I agree also.
Thank you. Sure, my screen jumps around. Kesham, please explain one mechanism of insecticide resistance at neurological level. Kesham, I think we tried to explain that, you know, how that works in our first tutorial, the broad concept. And I'm going to ask, I'm going to ask if you want to know more detail about that, read up about that. You know, just Google mechanisms of insecticide resistance and read up about it. It's fascinating. It's very, very interesting. But, you know, I don't think we really have the time to go into that level of detail at this point. Sorry about that, Kesham. But please go and read up about that. So at what level, Jivani Harishandra asks, at what level of resistance should we stop using that particular insecticide or that class? That's the whole point of these insecticide bioassay tests, is they use the WHO. So the insecticide manufacturer produces an insecticide, and through exhaustive tests, they come up with, through many tests on laboratory mosquitoes, wild mosquitoes, and so on, their product that they want to market and make commercial, they define the dose as defined by the manufacturer. Correct me if I'm wrong, Professor Terapap. They say the dose that you must apply this insecticide out in the field to kill 100% of mosquitoes is so many milligrams per liter of water, or whatever the solvent is that you're dissolving the insecticide in. WHO then says, okay, the manufacturer says that, let's say 5 milligrams per liter, it's going to be less than that, will kill 100%. So we are going to make test strips at 10 milligrams per liter. That's double the dose. And if 1 or 2% of the mosquitoes die, it shows they are still susceptible to that. But if more than that die, then you start getting resistance. And if more than 10% die, then they're fully resistant. So the level of resistance is defined initially, or the appropriate dose to kill your average susceptible mosquitoes is determined by the manufacturer. They work it out exhaustively. And then WHO develops those tests, and if your susceptibility test shows resistance, then you must stop using that insecticide. Prof. Terapap, any comment?
Yes, I think what you explained is good. Once you have seen the resistance happen in the population, so you have to find another, I think, another strategy to do. If you keep using the same insecticides, you will put more pressure into the same population, and then the resistance ratio maybe getting higher and higher. So I think once you see the population starting resistant, you have to do something. This is my answer.
Thank you, Professor Terapap. We're getting close to the end now, very nearly there. Here's another question from O Singh. Do we consider unable to fly mosquitoes as alive or dead, or for calculating percent mortality in a WHO bioassay? So knockdown is defined as a mosquito that is no longer able to walk properly, certainly not fly. It need not be dead. It just must not be able to to survive effectively. If it can't stand up and walk properly, and if it can't fly around anymore, it's no longer an effective vector, and therefore it's counted as a knockdown mosquito, and it's interpreted as that part of the sample that contributes towards resistance, it's susceptible. So if it's knocked down, meaning it can't walk properly or fly, it's susceptible to the insecticide. Gerard, comment on that?
Yes, so unable to fly is dead or live is depend, like if like the standardized of this, like if they are unable to fly at 1 hour, we consider like knockdown of mosquito. But after 24 hours, if they are unable to fly but they not they are not die, so it is a live mosquito after 24 hours. But if when 1 hour they are unable to fly, so we recognize that is a knockdown mosquito.
Okay, thank you, Gerard. Jeshi Doji, second last question. Is it acceptable, Gerard, I'm going to ask this to you.
Yes.
Is it acceptable to use mosquitoes that were collected from a cattle-baited net collection? Yeah.
It's like if I recommend it, depend on the situation, but it's back to the main concept that you have to know what is the main vector of your area that you have tested. If the main vector, they are also bite on cattle, and you can use cattle to catch that mosquito, is acceptable to test it. But if you are focused on like the main vector, but in your area that main vector they do not bite on the cattle, so you test with the other mosquito that they not transmit malaria. So it's like it's not useful for you to cattle bait. That is my opinion. Yeah.
So the important thing is you want to find out if you know that Anopheles dirus is your main vector in a particular area. You just want to catch Anopheles dirus from the area in which transmission is taking place, and if it's and if Anopheles dirus bites both humans and cattle, so if you can get Anopheles dirus off cattle, that's fine. That's no problem. But you're not going to use that specific individual. You're going to get the F1 generation from that. So I don't think that is important. How you could have collected them from larvae as well. So it doesn't really matter. So here's more questions coming up now. How can we interpret data with more than 98% mortality in repeat test? In other words, first test is 90 to 97%. Do we need to test again? Not sure I understand that. How can we interpret data with more than 98? Prof. Tirapap, do you understand the question?
The more than 90% mortality is considered susceptible, actually, because it's almost 100%. I don't know, first test is 90 to 97%. Maybe two different tests are different, maybe different test result. The first test, they got 90 to 97% mortality, right? And the second test, mortality higher than 98%. I don't know in this case, but actually when you do the test, you have to have the control. So see the control. If your control is less than 20%, it's okay. So I don't know. Maybe I'm not quite sure.
Yeah. No, that's fine. I think we need to make this one now the last question, because otherwise we're losing the number of participants.
Okay.
We're already down to 49, which is still good, and we appreciate all of you staying on and asking questions. That shows strong genuine commitment and interest, and we appreciate your interest. But this is the last one for today. When we collect the mosquitoes from the field, normally it was a mixed population that we collected. What percentage of individual species can be used for field collected mosquitoes? It is very difficult to identify the individual species. Yes, it's difficult to identify the species. It's not impossible. But the important thing is, you must know the identity of the specimens you are testing for susceptibility or resistance. It doesn't help if you catch a bunch of mosquitoes and you know, you do the test, you don't know if Anopheles diarrhea was in that sample. So, you know, you just catch mosquitoes randomly and you expose them to the insecticidal paper, they all die. It doesn't tell you anything, really. It tells you something, but it doesn't tell you anything that can help you in your vector control program. You must know that the identity of the mosquitoes that you, and normally what you would do is you would catch a bunch of mosquitoes in the field. You take each individual mosquito, each one live, and you put each one in a little tube with a moist bit of filter paper, and you get the female, one female per tube. That female lays eggs on the filter paper, moist filter paper. You rear those eggs separately, not contaminated with other individuals from another female. And then you take that female that lay the eggs and you do a PCR test assay on that female, or under, first of all, microscope identification. And if it's a part of a species complex, then you do PCR. And then you will know what is the identity of the larvae that came from the eggs that female lay eggs that you got the larvae from. And from there, you can start mixing the different larvae together and getting a bigger population. Once you know what was the identity of the female that lay the eggs that gave you the larvae that you are now starting to mix. But every female that you catch in a sample from a light trap aspirating off a cow or whatever, you need to put each female in a tube and get eggs from that female and keep those larvae separate so that you can identify the female, so you know what species are those larvae and the adults that will. Prof. Therapap, do you want to comment on that?
No, I think you are well now. Yeah.
Okay. All right. Prof. Therapap, Gerard, and our team, Dr. Tin, Dr. Porn, Wulan, everybody that took part in supporting this presentation. Dr. Rachadawan, I'm sorry I didn't ask you to respond. Dr. Pradeep, if you're listening in, to all of you, sincere thank you. Most of all, to our participants, sincere thank you for your interest. Good questions. We hope you enjoyed it. And tomorrow, same time, we'll have another episode. So thank you all for your time. We appreciate it. Thank you and bye-bye now.
Thank you and bye-bye too.
Thank you.